Well, welcome back. I'm glad you're all here and alive and mostly hale. Always a bit of a question when these things happen. I'm going to start by just touching on a couple of the slides. I'm not going to go through everything we went on Friday. That wouldn't make much sense. But it has been over a week since we've discussed it. So even for those of you who were here on before break, may not have things completely fresh in your mind before we go on to the ATP synthesis phase of oxidative phosphorylation. So I'm just going to touch on a handful of the concepts that we discussed the Friday before break, and then we'll get into the chemiosmotic side of it and how the ATP synthase works. So oxidative phosphorylation as a whole, taken as the entire thing, is the combination of electron transport using electrons from a variety of fuels in order to move protons, and in the process to create a proton gradient. And that proton gradient is, as we're going to see very shortly, the basis through which the mitochondrion is actually able to make ATP itself. And so what this makes possible in terms of this high-level map of everything is that many, many different fuels, carbohydrates, amino acids, fatty acids, can fuel down or funnel down into a common electron carrier, into NADH or in some cases to QH2, and that as those electrons are transferred to oxygen, the proton gradient is made, and then we're going to exploit that proton gradient in order to actually make ATP. And to flip rather quickly through these things, so we're going to be talking again about the mitochondrion. This is slide 3, I think. You have a double membrane compartment. There's an outer membrane and an inner membrane. We've talked about the citric acid cycle that's in the matrix of the mitochondrion. And then the electron transport chain and the ATP synthase are going to be embedded in the inner mitochondrial membrane. And this is an extremely schematic version of that inner mitochondrial membrane. That variety of electron carriers are going to move through complexes that are integral membrane complexes, ultimately transferring the electrons to oxygen. So burning the common fuels through oxidative phosphorylation generates water and carbon dioxide. The carbon dioxide came off in the citric acid cycle. The water is produced at this stage. Tied to that is the movement of electrons. So a gradient of more protons outside, tied to the electron movement is a movement of protons. So over the series of electron transfers, a proton gradient builds up outside the inner mitochondrial membrane. And then we're going to see how letting those protons escape or run down the gradient is going to drive the synthesis of ATP. And we took a look at a few, this is slide 8 here. We took a look at some of the common redox pairs of compounds that are either the oxidized or the reduced forms of things. At this end of the scale, the reductant is good at giving up electrons. So alpha-ketoglutarate will readily give them up. Sorry, this should be panel 8, I think, yes? Okay. You'll notice here that NADH and NAD+, NADPH, NADP+, are identical in terms of their redox potential. So they're not going to differ in how well they give up or pick up electrons. They're used in separate ways because that phosphate acts as a sort of a marker on NADP. And then if you notice, as we go down this series into the things where you have stronger and stronger oxidants, we're actually moving down some of the components of the electron transport chain of oxidative phosphorylation. So you have cytochrome B, underneath it ubiquinone, cytochrome C, and ultimately oxygen. Oxygen is an amazingly good oxidant, no surprise. We're going to see there is a better one, very unique one, but it is just about the best. And then figure 10 is the overview that gives you a very high order picture of the components of the electron transport chain. Remember that all of these complexes, complex 1, 2, 3, and 4, are embedded in the membrane. They are membrane complexes. And there are multiple proteins, all of them embedded in the membrane. And NADH will transfer its electrons to complex 1, which has its name of NADH-Q oxidoreductase. It's going to be catalyzing redox reaction between NADH and Q. Q becomes the lipid-soluble electron carrier that flows inside the hydrophobic interior of the inner membrane. Reduced Q can also be produced at complex 2. We met that before. That's a member of the citric acid cycle. It's the only component of the citric acid cycle that's a membrane protein. But the Q is going to carry electrons to complex 3, wherever it got its electrons from. At complex 3, the electrons are transferred between Q to cytochrome C. So that's why it's another oxidoreductase. Cytochrome C is a peripheral membrane protein. It's going to roll along the surface of the inner mitochondrial membrane and carry electrons to the next and last complex, which is complex 4. And complex 4 is cytochrome C oxidase, and it is the component that's capable of transferring electrons to oxygen, this incredibly explosive, flammable, poisonous, nasty compound. You don't think of it that way because it keeps you alive, but it is a very poisonous and nasty compound. So these are the major components of the electron transport chain at each of these, many of these steps. So in complex 1 and complex 3 and complex 4, that transfer of electrons is coupled to moving protons. So at these 3 steps, you're going to have more and more protons building up outside the inner mitochondrial membrane. This is sort of the Cook's tour of mitochondria. This is complex 1. I want to just remind you what it looks like because we're going to see a comment made about this when we at the end of the other slides. So this is complex 1 that can take NADH and transfer its electrons to ultimately to Q. This is just a reminder, figure 23. I'm not going to go through all the electron transfers here, but just to remind you that oxygen is a terrible, nasty, horrible compound. Once oxygen gas, O2, enters the complex, it is always attached to something. So it comes in as O2, and you can see it here in a variety of oxidation states, but it's going to remain attached to the complex until it leaves in the form of water, because all of these other oxidation states are things that can oxidize other things, and we don't want to do that unless we can get energy out of it. Okay. This is just a reminder that the potential energy drop at each of the 3 major complexes, 1, 3, and 4, is quite substantial. Each of these has a large enough free energy change associated with it that in principle you could make an ATP there. We didn't. All we did was move protons. But we had a big enough energy drop there that we had the equivalent of at least an ATP. We may have moved 3 or 4 protons, but we had the equivalent energy of an ATP or more. Okay, so now we're back to where we finished up on the last day before the spring break. So we've spent a fair bit of complex thinking and design and energy into moving these electrons from a higher to a lower level along that electron transport chain. We've moved protons at each of the 3 major complexes, not complex 2. And so what we've done is to generate a chemiosmotic gradient. And so if you take this as a schematic for that, you have the matrix, which is the innermost part of the mitochondrion, and you have an intermembrane space that's between the inner mitochondrial membrane and the outer mitochondrial membrane. So that's the intermembrane space. And each of these complexes, 1, 3, and 4, have been blowing protons into that space. We call this gradient of protons an electrochemical gradient. It's an electrical because there's a difference in electrical charge across that membrane. but it's also a chemical one because there's a difference in concentration of actual stuff. When we move 2 electrons from NADH, we move roughly 10 protons because we can do it at several points. So we move several at 1, several at 3, several at 4. All of this was the insight of a man called Peter Mitchell. And Peter Mitchell was a brilliant English scientist who was able to see past the groupthink that was going on when mitochondria were originally being studied in the 70s. Everybody wanted to isolate some high-energy compound, like phosphoenolpyruvate, that could make an ATP. And people invested incredible effort trying to find those high-energy compounds. Peter Mitchell's insight was that oxidative phosphorylation is the property of the whole mitochondrion. There's no one piece of it that by itself can accomplish it. But instead, it takes this intact mitochondrion that can generate a gradient and then exploit the gradient to make ATP synthesis. So a brilliant man. He spent his personal fortune on investigating this. The other thing that turns out to be important about his insight is that the chemiosmotic gradient is not just used to make ATP. It's used for photosynthesis. And we're going to see how many of the characteristics of this process transfer to photosynthesis very shortly. It's also the way in which bacteria that have a rotary flagella drive their flagella. They can use the proton gradient directly. They don't have to take ATP to do it. They use the gradient. So very, very clever device. So how do we do that? How do we do that? Well, there's this very nifty molecular machine called ATP synthase. And ATP synthase is able to use that proton gradient in order to make ATP. It can be divided conceptually into 2 major parts. There's a part of it that's embedded in the membrane. This is the inner mitochondrial membrane now, which we call F0, and it's an embedded membrane complex. And then a more peripheral part that's been called F1, kind of like the lollipop head on a lollipop. And F1 is the component of ATP synthase that's able to use the flowing of the protons and the rotation generated by that to actually generate ATP out of ADP and PI. So we can assign the proton flow, so to speak, to the F0 part and the ATP synthesis part to the F1 head. But they work together, and we'll see just how tightly together they work. Sorry. You can tell I was a little sick on break. I know. Sucks. All right. ATP synthase is a brilliant molecule, and its structure is quite generally conserved. We find it basically among all organisms that are capable of oxidative phosphorylation. And I'm presenting this to you in 3 different representations. We have a diagram over here. We have an actual electron micrograph here. So this is showing you that you can actually look at these things and see them. And then this is a space filling diagram from the structural studies based on purified proteins through X-ray crystallography and methods like that. So when we look at the F0 component, I mentioned that it's embedded in the membrane. Almost all of it's embedded in the membrane. There is 1 copy of A, and then 9 or more copies of this pink protein called C. And you can see that the C proteins have linked up together to make a ring. And whether it has 9 or 10 or 11 or however many copies of C that species has, all of them will always pack together to make a ring that's in contact with A. And then there are 2 copies of B that kind of sticks out of the whole plane of the membrane, and we're going to see that it is part of the machinery that communicates movement or makes it possible. So this is the F0 part. It's all in Roman letters, A, B, B, and many copies of C. The F1 component, that is actually where the ATP synthesis happens, gets Greek letters. And you can think of it as a trimer of dimers. That is, you have alpha, beta, make a dimer, 2 polypeptide subunit, and there are 3 of those. So you have alpha, beta, times 3. And then you have a few other components. You've got a delta that sort of sticks up at the end of B, and you've got a gamma that's sticking into this like a spindle. I'm probably showing my age here, but in the old days there were spindles that stuck in between records on record players and that kind of thing. And then epsilon that tweaks things a bit. So this is it as a diagrammatic form, and you can see in this space-filling diagram that this diagram is actually fairly truthful to the reality of the protein itself. So let's start by looking at what's going on down in F0 in the membrane itself. And a key part of it is that the C ring, the ring made up of 9 or more C proteins, is going to rotate. It's actually going to turn in the plane of a membrane. It's going to turn in the plane of the membrane, and it's going to rotate past the A protein, which will not move. So A is stationary, and the C ring rotates. And because these binding sites for protons here in the C ring are embedded in that lipid interior, they do not have access to proteins except when they're in contact with the correct part of A. So A provides a place for protons to enter the F0 complex and a way for them to bind to binding sites on the C ring. And there's a carboxylate residue there that's conserved. And so a proton can come in from the intermembrane space through the A protein, bind at that site. The previous sites have been occupied. The ring is turning and turning and turning and turning and turning. And when it gets around to this site on A, that provides a channel for the proton to come off and re-enter the matrix of the mitochondrion. The fact that there's a gradient here is going to favor it turning in one direction. If there was no difference in proton concentration, this thing would just kind of wobble back and forth. But because there's a vast excess of protons in one direction than the other, it actually makes it go in one direction. You can compel it to go the wrong direction by changing the directionality of the gradient. Any questions about this before we move on? Well, so far we have used a proton gradient to turn something. You can think of it, if you like a water wheel, where a brook flowing past the water wheel will turn the water wheel. But a turning water wheel does nothing unless you couple it to something else. So somehow we have to couple the rotation of the C ring to that alpha beta trimer to somehow or other make something else happen. Well, the alpha beta trimer, here you can see it in blue and green, does not move. It is stationary. And in fact, it's held stationary, if you remember, by the B and the delta subunits, right? So the alpha beta trimer is held stationary. However, in the middle of this, you notice a little purple bit, and that purple bit is the gamma subunit that was coming up from inside the C ring. If you go back to this diagram, you notice here's the C ring, which we've got spinning around because of the proton gradient. And sticking up out of it is this gamma protein that sticks up like a spindle. And it's going to stick up right into the middle of the alpha beta trimer. This is looking down on top of it. You can see the gamma protein here in the middle of the trimer. This is looking at the side of it in a ribbon diagram. And you can see the gamma subunit sticking up through the middle of that sort of very tight donut, and here are the alpha beta subunits. Okay? Now, as the gamma turns, when the C ring turns, the gamma turns. And as the gamma turns, it's going to touch those alpha beta dimers in different ways. So the turning of the gamma is going to trigger changes in conformation in the alpha beta subunits. And this may be a little clearer in the next diagram. So here we're looking down on top of F1. We're looking down on top of that alpha beta dimer. And we're going to look at the 3 different states that the subunits can enter into depending on how they're interacting with the gamma. So here the purple elongated triangle is the gamma subunit, sticking up, turning with the C ring. And what we're going to see is that the alpha-beta dimers can take on any one of three different states called open, tight, and loose. And which state they're going to be in is going to depend on how they're interacting with the gamma. So let's just start here for the heck of it, all right? And here we have the top subunit is in the open state because it's got the pointiest bit of gamma touching it. And in the open state, it's free to let an ADP and an inorganic phosphate bind at that site. When it is in the open state, its neighbor here is in the tight state occupied by an ATP, and its neighbor over here is in a loose state occupied by an ADP and PI. Now, the C ring is turning and turning. It's going to force that gamma subunit to turn, and when it does that, the ADP and PI that came into the open state is now here in a loose state. It's in the two pieces are there, but nothing's happened yet. On the other hand, when the gamma spindle pokes this subunit, it forces it into the open state, and the ATP that was sitting there in the tight state is now released. The third subunit that had the ADP and PI bound to it, when that rotation happened, got squinched, and now we've squinched the two pieces into one ATP molecule. We have another turn of that rotor. That rotor will now poke this subunit to open up and release its ATP, but this one has been reoccupied by inorganic phosphate and ADP, and that one was squinched enough to make an ATP. And with one more turn of the spindle, the ATP that was formed at that site is released, and we're ready to do everything all over again. So it rotates in sort of quanta. The C ring is moving with its protons, but the gamma spindle kind of jumps from one to the next to the next. And each time it twitches or turns, it's changing its interactions, poking at the alpha-beta dimers in different ways, and forcing a conformational change in them. Questions? This is really very sly. The F0 is actually rotating from the flow of protons, but because it connects to the F1 through the spindle, it can turn that into pokes at the subunits that then triggers changes of conformation that drive the formation of ATP. Okay. So the kinds of interactions we've talked about so far, it should be very clear to you that it is critically important that intermitochondrial membrane is an intact lipid bilayer which does not have holes in it. Because if the electron transport chain is moving electrons from NADH to Q to cytochrome C and eventually onto oxygen, the protons that it's moving have to stay on the side of the membrane that they are put on. This whole process of oxidative phosphorylation depends intimately, absolutely, on the integrity of the mitochondrion as a whole, because you have to keep those protons where they are. If there's any way for those protons to slip back across other than the ATP synthase, you're going to lose the energy that was accumulated in that chemiosmotic gradient. Another way of describing this connection between electron transport and ATP synthesis is we say they are coupled. There's a coupling of oxygen consumption and ATP synthesis. And they are coupled because the proton gradient is the thing that's shared by both of them. So if there were some way that you could, in fact, make holes in the intermembrane, intermitochondrial membrane, or if there were some way that protons could get back into the matrix other than through the ATP synthase, we would then say that there's been uncoupling. There's no longer a tight connection between electron transport, oxygen consumption, and ATP synthesis. And there are a number of molecules that are known to do that. It makes predictions. It predicts that if you could somehow do this, you should be able to stop making ATP out of the proton gradient, and so on. And this is one of those compounds. This is called dinitrophenol. And if you look at this, you'll notice that dinitrophenol has the capacity to be protonated here. And look at these two forms of it, and which of those forms do you think would be the most readily able to go through through a biological membrane? There's some charge on this one. This is all neutral. NO2 groups, nitrogen and oxygen, are fairly close in electronegativity. So an NO2 group is not a very polar group. And so the protonated form of the dinitrophenol fairly readily passes through a membrane just by diffusion. It does not need a carrier. It just simply diffuses through the membrane. And on the other side, if there's not very many protons on that side of it, it can release that proton. And this form is capable of going through as well. It's just not quite as great at it. So what this means is, since it exists in two forms and can pass through the membrane without an actual carrier or channel or pump or any of those things we talked about in the membrane chapter, it means that if there were a lot of protons on one side and you put in some dinitrophenol, fairly rapidly there would be the same number of protons on both sides. And so we call dinitrophenol, or DNP, an uncoupling agent. It's capable of uncoupling electron transport and oxygen consumption from ATP synthesis. In the presence of DNP, you will still see electron transport, you will still see consumption of oxygen, but you will not see the production of ATP. So where do you think the energy is going? We mentioned that there's roughly an ATP worth of energy involved in each of those three electron transport steps. What do you suppose is happening to that energy when you uncouple electron transport from ATP synthesis? There's sort of a default setting for garbage energy. You want to take a stab at it? Heat, exactly. When a system doesn't know what else to do with energy, it gets degraded into heat. And so when you have an uncoupling agent around, you will not be producing as much ATP, but you will instead be generating heat at that site. I've described this as a external reagent that might be used by experimentalists. It was used, people attempted to use it as a weight loss aid in the 20s and the 30s because, well, if you can make the mitochondria use up energy without actually getting anything out of it, maybe we can lose weight. Well, they did, but they also lost their kidneys. So not such a good one. Worse things have been done. Ask me sometime about tapeworms. You could get them in jars in the 19th century. Yeah. But what's interesting is we have proteins that actually do this on a regulated basis. We have things called uncoupling proteins. Uncoupling protein 1 and uncoupling protein 2, really imaginative names. And the reason we have them maybe is kind of obvious if you think about it. You and I have body temperatures around roughly 37 degrees right now. The air is not 37 degrees. We're getting that heat from somewhere. And one of the ways we do that is by a controlled amount of uncoupling of the mitochondrial membrane. It's interesting to note that during pregnancy, the metabolic changes that happen to women change in many women the levels of uncoupling protein, so they do actually become more efficient. It's like nature's against us. But that's one of the ways in which the metabolism of pregnancy can tweak the efficiency of the body. So this is repeating some of what I've said here, that you have dinitrophenol here and protonation, and that this is capable of uncoupling electron transport and oxygen consumption from ATP synthesis. We've talked about physiological and drug ones. One of the places in which very intentional uncoupling goes on is in a special kind of fat tissue. If you look at most of the fat tissue in our bodies, it will look pale white to yellow. If you look at certain kinds of fat tissue in animals like bears, other animals that deal with the cold, they have brown fat. And the brown fat is brown because it has a lot of mitochondria. Our fat doesn't have mitochondria. It's just a storage depot for the most part. But brown fat has mitochondria right there next to the fat because it's a place where the body can deliberately uncouple mitochondria and generate heat for the use of the body as a whole. And we have a little picture here of a hibernating bear because the bear is so extraordinarily good at getting through a winter without freezing to death and making the best use metabolically of what she's got going. I just want to sort of touch a little bit on sort of the metabolic amazement of a hibernating female bear. She can regulate her body heat through these brown fat deposits that can be delicately tuned up or down with uncoupling protein in order to make more or less heat right there. Typically, a hibernating bear will be pregnant, so she's going through the metabolic demands of a pregnancy doing this. She will develop the first level needed for lactation so that when spring comes and the cubs are born, she can start to suckle. And to do that involves retooling an extraordinary fraction of metabolism. A hibernating bear does not burn sugar. She only burns fat. So we're going to see in a little bit about fat metabolism and beta oxidation, but fat has twice the energy density of sugar. So by burning fat, she's getting twice the buck per gram, twice the bang per gram for energy production. The other useful thing about burning fat is that the byproducts of that are water and carbon dioxide. So she can breathe out the carbon dioxide, and the water she produces by burning her fat provides most of the water needed during hibernation. You may remember that adipose tissue has triacylglycerides, so there's 3 fatty acids plus a little glycerol backbone. The female bear uses that glycerol backbone as a way to capture nitrogen that might be around, so she can make amino acids using the glycerol backbone. I'll be posting on an icon shortly an article about how the hibernating bear has almost no protein loss. So there's something in hibernating bear serum that turns off protein degradation in, for instance, a mouse or something like that. So there's this ability to readjust major metabolic demands through this whole process. And she'll come out of the cave 40% lighter than she went into it after basically every system being tweaked. Any questions about oxidative phosphorylation or ATP synthesis before I cover the couple of comments and questions that we got on Friday before the semester? So don't move yet. But do you have any questions about the things I've said so far? Okay, let me just close that one for the moment. And here we can go to this. I'm going to post this. I'll be posting this little small PowerPoint. You don't have a handout for it yet. I'm sorry. On icon, so if you want to look at it in more detail, you can. This is just another diagram of the electron transport chain, showing it going from NADH. Here's your lipid-soluble Q, your next component of it, your peripheral membrane cytochrome C, cytochrome oxidase using oxygen to make water, and then the ATP synthase using that proton gradient to make the ATP. Among the comments that I received on Friday was that here is the ATP, here's complex 1 of the electron transport chain, and someone noticed that it looks like a crawling baby with a really big head. I am never going to think of complex 1 the same way again. So there you go. Who knew? You know, all kinds of detail in there. Someone asked, Will extra credit from the exam that boosts the grade up to 80 plus also mean that it is now an A? And just to clarify how that all works, the bonus credit from the little essays gets added into the course total, mechanically speaking. But if you find it useful to think about adding it to a particular test, feel free to do that. It has the same effect either way. Someone asked, what are the differences between the complexes? They're called complexes for a reason. They have many proteins in them. They're going to differ in which electron carriers they use. They're going to differ in their redox potential, how good or bad they are at pulling electrons from things, and at how many protons are moved, if any. Complex 2 does not actually move protons. Someone asked, is that a Jetta that I drive? Yes, it is. All right. Someone asked, I answered.